seminar. So today we have Professor Leslie Yale, Professor of Chemical Engineering from RMIT University in Melbourne, Australia. So he is a world's leader in a lot of topics. Above all, he is the editor-in-chief of Biomicrofluidics. I also have to declare a conflict of interest. I'm an associate editor. So it's great to have Leslie here to share with us on this topic, sonomechanobiology, a new field of high-frequency cell mechanostimulation. For those of you who may not be very familiar with Leslie's work, he has been working a lot on the acousto-microfluidics and a lot on fluid dynamics and how sound and acoustic interact with microfluidics. And this is a new area, and I hope our staff and students and colleagues can get more information and inspiration from this. And a lot of colleagues told me that they wanted to come, but because of clash in some of the lectures, they will not be able to come. But I think the message will spread across the campus. Yes. So without further ado, please join me to welcome Leslie.
Thanks, Anderson. Thanks. Oh, yeah, that's true. Okay, so thank you very much, Anderson, and it's great to be here. I used to come to Hong Kong quite a bit, like 2-3 times a year, but that was before COVID. So this is actually my first time back to Hong Kong after COVID, so it's always nice to be back. Let's see. Okay. All right, so by way of introduction, I'm a chemical engineer by training. And then I did my degrees in Imperial College in London, moved to the States where I did a postdoc, and then after that, I started off my career in Monash University in Australia, where I was in mechanical and aerospace engineering. And then I moved to RMIT, where for some reason, they put me in electrical and computer engineering. And then they discovered that I couldn't teach anything other than electrical circuits, so I moved to chemics. Back then, it was civil, environmental, and chemical engineering. And now it's mainly just chemical and environmental engineering. But so, what does that make me? I guess confused, right? But anyway, it's great to be here. Let me just tell you a bit more about our work. But before that, if you haven't been to Australia, come and visit us. We're in Melbourne, so that's right at the bottom here. It's a big country, but Melbourne's really beautiful, so come and pay us a visit. If you are old enough to remember this television series called Lost, I don't know anybody. You guys are so young, right? You don't remember. So Lost is a television series. I think I was a postdoc when I was watching it or something like that. So it tells you, probably a long time ago, it tells you how old I am. It's about a plane crashing in some Pacific island, and they were, stranded and all. But one of the things in one of the television, one of the episodes, they said, they were talking about Australia, and it's called Down Under, right? So, and they said, the reason why it's called Down Under is because it's the closest place you can get to hell without being burnt, right? So we're pretty close to hell being at the bottom of the country, except for Tasmania, which is really hell because they got the Tasmanian devil, right? Anyway, so these are the people who do the real work. I just get to tell you about it. And all the work that I'm going to tell you is done single-handedly by one person, and that's Liz. She's a brilliant, Liz Bona-August. She's a brilliant cell biologist. She teaches me cell biology, all right? So all the credit is to her. Okay, so this slide probably summarizes all the work that we do in our lab. So we we're a bit of a jack of all trades. We do a lot of things, and maybe to our detriment. But core to everything that we do are these high-frequency sound waves. Okay, so by high frequency, you can just remember these magic numbers. What I'm talking about is 10 megahertz. Okay, 100 micron wavelengths, 10 nanometers in amplitude. Okay, so it's really multi-scale. Okay, and so people have done ultrasound for a long time, but that's down in probably hundreds of kilohertz. We're playing up in 10 megahertz. And when I started my career, it was mainly in microfluidics. And we started coupling these high-frequency sound waves into fluids. I guess nobody's crazy enough to do these things, so we see all kinds of interesting phenomena that we couldn't explain. So for first 10 years of my career, I started pulling all my hair out because we see all these things that we couldn't explain. So it took us a long time to actually build the theories because all the classical theories didn't apply, right? Especially when you go up in frequencies, everything becomes very nonlinear. So all the linear theories that were all, like classical aeroacoustics, they couldn't explain what we were seeing. So we had to, start from scratch and all. So that's, 10 years of work. We try to, not just do fundamental work, but also translate it into real-world microfluidic devices. And then after this time, we sort of thought, oh, we kind of understand a bit more now about this high-frequency acoustofluidics. So I get bored, right? So then we couple these things into materials, 2D materials and bulk crystals, and then we see all the crazy things happening again. So it's a bit of deja vu. We're starting to piece together what's happening. Same thing with cells and tissues. So I guess this is the story of my life. I get into things that are more trouble than I can handle. So I'm going to tell you a bit about these for the rest of the talk. Now, what are those sound waves? Okay, they're called surface acoustic waves. You probably use them every day of your life, but you may not know it, right? Your mobile phone has 4 or 5 of these devices, and they've been used for signal filtering in telecommunications for about 60, 70 years now. So it's old technology, but it's, like I said, it's mainly telecommunications. Very few people play with fluids or materials or cells, okay? How we generate these is on a chip, right? So this is a piezoelectric material, and you pattern these interdigital transducers with just finger-like electrodes here. And just like the shifting of tectonic plates gives rise to an earthquake, okay, you put an AC signal, because it's a piezoelectric material. Piezoelectric material converts electrical energy to mechanical strain. So you put an AC signal, these fingers go back and forth, and how an earthquake is generated, you launch this nano earthquake, right? 10 nanometers in amplitude, 100 micron wavelengths. So that's the rough order, okay, 10 megahertz. All right, so we've also discovered it's not just surface acoustic waves that matter. So a pure surface wave is on the surface, all right? So if you look at the chip, okay, you scan the chip, here's the nano earthquake. On the back side of the chip, you don't see anything, all right? On the flip side of it, you've got bulk waves, okay, where the energy goes throughout the whole chip, the thickness of the chip. So you see a very different pattern. You see it on both sides. When you match the wavelength and the thickness of the material, something weird happens, which nobody looks at, okay? Bulk waves are known, for a long time, okay? And just like I'm speaking to you, bulk waves go everywhere, right? It's a 3D phenomenon. Surface waves are localized on one surface. When you've got this matching, this, in this asymptotic regime, where you get this matching, it behaves really weirdly, right? So, here's a simulation. It's a bit upside down. But if you start off with a surface wave, like how we generate these with the interdigital transducers. It starts off the surface wave, but the energy leaks all the way to the backside because you're matching the wavelength and the thickness, right? So it goes and reflects off. So it's neither a bulk wave or surface wave. It's this weird hybrid. Now, when we discovered this, I guess nobody talks about this. So there was no name to name it. I could have called it Leslie waves, but then people stoned me for heresy. So we call it surface reflected bulk waves, which on hindsight, now we have published the paper, you can't change it, right? But now on hindsight, I think surface reflected bulk wave, it doesn't mean anything, but here we are. We're just stuck with that name now. But anyway, the phenomena I'm going to tell you, they bear, although it has bulk characteristics, it bears a lot of resemblance to surface waves. So we play around with both of these things, okay? The interesting thing, like I said, is we're up in this frequency, okay, about 10 megahertz. People do ultrasonic imaging at that frequency, but most of chemistry or fluidics has been all done at under megahertz. And that's because all the bulk waves are pretty much below megahertz that you can easily generate. All right. Now, the thing with the low frequency, as I said, everything's linear, but more importantly, a lot of it is driven by cavitation. Okay, and cavitation, if you know, is this really rapid implosion of the bubbles, okay, gas bubbles in a fluid. Okay, and when you have this really rapid implosion, it's very high pressures, almost more than gigapascal. Very high temperatures. And that tends to overwhelm everything. Okay, so you can do a lot of chemistry, you can do a lot of drug delivery and things like that with cavitation because there's so much energy, right? But that typically overwhelms everything. We almost always never get cavitation because as you go up in frequency, okay, the energy required to generate cavitation exponentially increases. Okay, so we don't see cavitation because on the chip it's very efficient, so we use very low power to begin with. So all the fundamental mechanisms for what we see is very, very different, okay, from low frequency. Okay. Now, most people assume that it's all mechanical driven, okay? And that's reasonable assumption, okay? Now, I've told you that the waves, the amplitude is 10 nanometers, but the frequency is 10 megahertz, okay? Now, if I tell you that if you're sitting on the surface of this nano earthquake, okay, and it's going up 10 megahertz, 10 million times a second, if I tell you the velocity is 1 meter per second, what is that acceleration? This is physics 101, okay? 10 million times a second, I'm going 1 meter per second, okay? The acceleration is 10 million Gs, all right? Now, it's no wonder why we see phenomena because, you're driving things at 10 million Gs. And so for a long time, everyone assumes that, it's purely mechanical. You've got 10 million Gs of acceleration, that's a huge amount of force, but because it's an electromechanical coupling, it's a piezoelectric material, you've got an underlying electric field, it's not just mechanical. We're beginning to discover that we see a lot of effects where you have the electric field in play as well. Static charging, dynamic charging, free radical generation, charge polarization, and things like that. To the point we can even split water with these surface acoustic waves, right? So, that's a story for another day, but I just want to get you to just appreciate that, the conventional mechanisms, conventional mechanisms of cavitation is not at play. There's some interesting nonlinear phenomena coming not just from the mechanical field, but also it's the electromechanical coupling. Okay, so bear those in mind. Okay, but for the rest of this, and that's why we see all these crazy things happening. Okay, you got these very nonlinear electromechanical coupling at play, which is rather complex. Okay, I must admit that we don't quite understand even most of what we see. Okay, so I'm sure there are very clever people in this room, so you can help us try to piece together some of the phenomena that we see. Okay, but let me just tell you a bit more about cells, because this is something new work that we've been seeing, and there's some interesting phenomena that we're trying to explain. Okay, so the experiment is very simple. Okay, you saw that chip, okay, and here are these nano earthquakes going along the chip. Okay, now we couple that into, say, a petri dish, or a well of a microarray, as you see there. Okay, and the cells are in there. Okay, so we, basically, as you see, there's a coupling, and it just vibrates the cell. Now, I must say that we use low enough power that we don't have flow. Okay, so there's very little shear, and that's deliberate because I don't want to complicate things. Okay, I want to remove the component of shear. Okay, so the cells are just gently sitting there. Okay, and in everything we do, we don't use any chemical factors and all. Okay, so everything you see here is just due to the vibration. Okay, gentle vibration. Heating we try to eliminate as well. So so that's important because we want to find out what the real driver is. Okay, and we don't want all these complicating effects. Okay, so what we see. Okay, at the beginning, we didn't know what we're doing, so we just threw a whole bunch of gold nanoparticles into the cell suspension, and then we looked at what happened. Okay, these are 10 nanometer gold particles. Compared to the control, okay, you see this is like 5 to 10-fold uptake of gold nanoparticles in just 10 minutes. Okay, same thing with dextrans. All right, you can have different molecular weights. Obviously, if you use higher molecular weights, the uptake will be lower. But even at 250 kilodaltons, you see a 2-fold uptake in just 10 minutes. Right? So what's happening? So we thought, maybe it's endocytosis or pores, because people have been doing sonoporation for a long time, right? And it's mainly driven by cavitation. They make pores. All right. We tried very hard to see whether there's any pores. We didn't see anything. There's some aberrations on the surface, but none like the large pores that people tend to see. Right. But admittedly, trying to see pores is very hard. So what convinces it's not pores is this, right? If you look at the calcium, when we turn on the acoustics, the calcium goes up. When immediately after the point where we turn it off, it shuts down, right? Same thing with propydium iodide. Okay, if you know propydium iodide, that's the marker for cell death, because propydium iodide goes into the cell, right? Same thing. You turn it on, the acoustics, it goes up, all right? The intake of propydium iodide, but then immediately when you turn it off, it shuts off. Now, to me, pores don't heal so fast, if at all, right? It takes many, many minutes or hours for pores to close, right? Not immediately. If you look at the flow, you see flow cytometry data, you also see it shutting off immediately. But what was more convincing was the FTIR, because that tells you the structural organization of the lipids, the membrane lipids. Right? So you turn on the acoustics, you see a shift in the membrane structure. When you turn it off, it's gone. Right? So something's happening. We're altering the lipid structure and permeabilizing the membrane, but it's reversible. It's just transient. Okay? It shuts down. It relaxes almost immediately, which is interesting because now we have a mechanism that doesn't rely on pores. And because of poration, and because of that, because poration, the cells really don't like it, right? And so your typical poration, if you work with sonoporation, is 60%, 70%, same with electroporation. Okay, we get almost 95% viability with the cells, okay, which is great. And then they continue to proliferate as per normal, right? We know it's also not endocytosis because we look at we use different endocytic inhibitors. There's no difference, and more importantly, because in endocytosis or endocytic uptake, things get localized in the endosomes, and then they get recycled through the lysosomes, right? We see things being distributed throughout the cytosol. Okay, so which is good, okay, because we're maintaining high viability but also getting cytosolic uptake. Okay, if you know anything about intracellular delivery, you know that there are two methods in general. One is these physical methods that actually make pores in the membrane, right? And that has the advantage of getting things into the cytosol and not localized in the endosomes. But cell viability is very, very low. On the other hand, if you use biochemical methods like a Trojan horse to sneak things in, like polymers and viruses, nanoparticles and all, right, your cell viability is high, but then everything gets localized in the endosomes. You need endosomal escape strategies to get things back out, else you lose all of them in the lysosome degradation process. Right? So in some ways, we have the best of both worlds because we have high viability, we have cytosol delivery. Right? And then if you can do that, then you can drive things through into the cell for all kinds of things. We did it with sRNA just to show. We see in 10 minutes we get twofold knockdown of the gene expression. Okay. Now, you can get things into the cell. You can also get things out of the cell, right? What we saw was that we can stimulate the cell to produce more exosomes. And if you work in exosome diagnostics or therapeutics, exosomes are really good for a lot of things. But then the problem is trying to get enough exosomes is a big problem. So we can stimulate the cells to get about 8 to 10-fold enhancement just by just in 10-minute cycles. You can stimulate production of the exosomes, not just release, but stimulating the cells to produce more. Okay, there's some interesting mechanisms for this, but it's all calcium driven. Okay, so just remember that for a while because I'm going to come back to this this theme of calcium. All right, so basically what I'm trying to get at is that when you stimulate cells, you can get them to do things, right? You can get things in, you can get things out, or stimulate more exosomes. You can also drive stem cells down a differentiation pathway. So with this 10 minutes of stimulation just for 3 to 5 days, okay, this is not continuous stimulation. There's no chemical factors at all. Just 10 minutes of stimulation, we can push them down the osteogenic pathway. And the interesting thing is we get very, very early onset of osteogenesis, okay, in about 3 days. And also, if you look at the late markers, you see that they persist for long term, okay? Keep this in mind because I want you to remember these two time scales, the early onset and the long-time persistence, because you'll see when I talk about mechanisms, these two things coming back into why we get both, all right? Which is very unusual. Okay, most people have done ultrasonic stimulation, for example, with stem cells. They continuously stimulate for many, many days, right? And they don't see this early onset. We just do 10 minutes of stimulation, just for 3 to 5 days, and we see this early onset, long-term persistence. Okay. Again, it's calcium-driven. Okay, so we'll come back to this theme about calcium. Endothelial cells. If you look again within, sorry, I'm a bit short. Maybe I'll use the pointer. Okay. So when you just immediately after 10 minutes stimulation, you see cytoskeletal rearrangement. You see these actin stress fibers that form, but within about 20 minutes, they relax and they're replaced by these circumferential actin bundles. The cadherin is interesting because these disrupted cadherin structures become more and more linear upon the in 20 minutes to an hour. Okay, so here is the two time scales coming into play again. Very, very early on, you get stressing of the cell. And this is the same as if you were to, stress the cell in different ways with chemicals, for example, with ionomycin or LPS or something like that. The cell gets stressed, okay? And what happens? It goes away from homeostasis and you push it down some apoptotic pathway, all right? Most people can see this, but then this longer time scale, they don't see. So we can challenge the cells, but also we see a recovery back to its original state. But the interesting thing with this recovery is that you get this linear junction arrangements, and what's the consequence of that is that the endothelial barrier is actually enhanced, right? So we can get 4 times. If you look under the TER, the transendothelial electric resistance, which is a measure of the permeability of these the endothelial junctions, we see 4 times increase, right? So which is great because if you work with organs on a chip, right? Everyone's trying to recapitulate, organs on a chip for, drug discovery and drug screening and things like that. The problem with all these things is that you can never get, the endothelial barrier to mimic what's in the physiology. It's about 10 times lower. So that's a big problem. Okay, so if we can increase the barrier function closer to that found physiologically, I think that will help with a lot of the current organ on a chip work at the moment. So in short, okay, I hope that I convince you that just this 10-minute stimulation, you see all kinds of different effects from driving things in the cell. You can modulate the endothelial barrier, differentiate stem cells. You can drive exosome biogenesis. Now, the big question is why? Okay. So, we don't feel 10 megahertz, right? Why would cells feel 10 megahertz? Okay, the time scales are all wrong, right? For example, the cells take time to respond, seconds, minutes, days even, okay? Lipid time scales, on the other hand, are picosecond order. So we're neither here nor there, right? The length scales, I just told you our wavelengths are 100 microns. The cell is much smaller than that. So you don't even deform the cell that way, right? So when we first started, we're thinking, what's the mechanism, right? Most people who do mechanobiology, well, they compress or they stretch the cell, they shear the cell, right? That's all static, or at best a few hertz, right? So nobody goes as high as megahertz. There's some people who play around with kilohertz, but they published this paper, okay? This is Matt Dalby's group at Glasgow. They're very well known in the mechanobiology field. They even said, in their paper, there's no benefit or little extra benefit in going beyond 1 to 10 kilohertz. And here we are 3 orders of magnitude higher, and we see all these interesting effects. So the question for us was, why? Right? And so we're trying to figure this out now. Now, one way we've done this is we try to look at the membrane tension. Okay, so the cell membrane is made of this lipid bilayer. There's a membrane stain called flipper, and flipper basically conjugates with the membrane lipids, and the conformation, it will change color based on conformation. In fact, its fluorescence lifetime is linearly correlated with the membrane tension of the cell. Okay, so this is well known. So we use flipper, and we see when we, at a certain power, you can see there's a decrease in fluorescence lifetime and hence the membrane tension. Okay? It's also you can also see a lot of the cell contracting and these Philippodia like protrusions, right? So there's basically a marked decrease in membrane tension. Okay, when we mechanostimulate the cells, together with the contractions, the protrusions. But like I said, just like you saw with the examples I gave earlier, the cell recovers after that within 20 to 60 minutes. Okay, so there's a recovery period. Okay, now what does this membrane tension tell us? Okay, so it doesn't say very much. So we went to look more at a different stain. We stained for lipid rafts. Okay, and you can see that with the mechanostimulation, you see an increasing order, or lipid order or packing. Okay, basically we are going towards a gel phase, right? But again, it recovers within that the 20 to 60 minutes. So, well, if you know about membranes, you know that you can have disordered lipids and you can ordered lipids, right? So these are the ordered ones are the saturated ones and the saturated cholesterol, for example, and then the disordered is the unsaturated. So you can have this phase condensation and phase separation. So this increased packing or order when we stimulate decreases the membrane tension. Basically, you get clustering of these ordered lipid structures. All right? And why is that important? Well, because when you cluster, you've got all these membrane proteins as well, membrane-associated proteins that get pulled. And some of them are mechanosensitive ion channels. Okay, so mechanosensitive ion channels are like piezo and things like that, are ion channels that open or are activated because of mechanical stress. So now when you pull on them, you see, I'll show you here we stain for both. We stain for, the purple is the lipid raft stain that I talked about just now. And we stain also for piezo, okay? And you can see here, here there's co-localization of the lipid rafts and the piezo. Okay, so blue is the control. This is the distance between the piezo and the lipid rafts. You can see when we stimulate, you see this red one, they cluster together. Okay, and over time they relax back. So what we're doing is, when you have coalescence of these lipid rafts, they pull on the piezo channels and they activate them. Right? So, and this is known. I mean, the fact that they are activated through a force is known in the the ion channel literature. Okay, so what I'm trying to get here is that when we stimulate, okay, you get this manipulation of the membrane lipids. They cluster the ordered ones. There's some ordering and clustering that then pulls on the piezo channels, activates them, allows calcium to go in, right? When you turn it off, it relaxes back, okay? Now, the interesting thing is that's not unique, okay, to this stimulation. Other kinds of stimulation can do that. What's unique here is that because when you push calcium in, okay, the cell doesn't like it, all right? So it's in its challenge state, drives it away from homeostasis towards apoptosis. Now, the beauty of what we are doing is because we use so little or gentle stimulation for so short periods, we don't kill the cell. We can moderate enough calcium to go in, a lot more calcium to go in, and it gets pushed into the ER. And over time, it slowly releases back. This is called calcium-induced calcium release. It releases back over time, back into the cytosol, back into the extracellular milieu. Okay? And this is the 2 time scales that I was talking about, right? You push things in, okay? And then over time, it releases back out. So that's why we have very early onset, for example, of the osteogenesis and long-term persistent effects. Okay? Same thing with the endothelial or the cytoskeletal rearrangement. You get this short-term effect of challenging the cells. You get this active stress fiber bundles, but then it releases back over time, and you see, the relaxation of the cell. You see increase in the endothelial barrier integrity. So I hope that, maybe it's not a complete picture, but I hope to convince you that maybe, we're starting to piece together the puzzle. Why do cells, how do they respond to 10 megahertz frequency, and why do we see what we see? Okay, so, and once you get calcium in, well, calcium is a second messenger, it's a universal second messenger, so you can then, trigger all kinds of secondary signaling cascades that then, push the cell in different fates, right? So, once you have calcium in, don't ask me about signaling pathways, okay? This is beyond me. You can do different things with the cells, all right, and this is what we see, the differentiation. We're looking at oocyte and sperm activation, neuromodulation, you can, as well as all this proliferation and migration. Okay, so, and some of that I've already shown you, okay? So I think, how much time I have? I should finish here. But I hope to convince you that, we're not just crazy, okay, well, half crazy maybe, but there's hopefully a new field or can of worms, depends on how you want to see it, but we've opened, and perhaps maybe this is it could be useful for some things. And we hope that there'll be more people that will come and help us to try to piece together and understand some of this phenomenon that we see. Anyway, this is a review paper. We've tried to put all these thoughts together. So thank you very much. Happy to take any questions.
I'm pleased to see you are working on endothelial cells. This is my area for many years. We're working on this impact of shear forces on the regulation of endothelial cell function. Basically, the different, in vivo, there will be the different pattern of blood flow.
Yes.
Like a laminar flow, probably and the disturbed flow, turbulent flow in the curvature of blood vessels. But here you use this acoustic cell stimulation, and then open up piezo-1 channels, allow calcium influx and trigger something. These are very interesting. And then, of course, you use this high frequency, because, for example, in our heart, because we are more interested to mimic the actual situation. Yeah. We have a project on the exercise. When you exercise, heartbeat increase, shear force increases, and they have a better impact in terms of producing more nitric oxide, all those things.
Yeah.
Now, for example, in your system, because those are high frequency, our cell never experience, for example. Yeah.
That's right.
And it's possible, because I saw your trip, you have this, frequency, because if our heart beats once per second, that means 1 hertz. Yeah.
Yes.
If you're doing exercise, your heartbeat can be 3 times higher.
Yes.
Might be 3 hertz. Yeah.
Yes.
Whether or not in your system can design this, 1 hertz and 3 hertz, and then this wave and acting on endothelial cells, monolayer, and then detect certain things. For example, whether or not now you higher shear force, normally we believe to increase production of nitric oxide, for example, yeah,
Yes.
or other things. Possible in your system, you can do this, and then that data will be interpreted to, for example, in what level of exercise you need to achieve,
Yeah.
and produce a health beneficial the impact on
Yeah.
the vascular function.
Yeah. So you're right. I think a lot of people in mechanobiology are doing this. It's easy to do, 3 hertz or something like that, right? So they study that. My system cannot go down. Okay, my system will never go down below a few megahertz. Right? So it's not useful for what you're saying. And this is, in some ways, you're right. You touch the hot potato, right? Every reviewer always comes back and says, where's the physiological relevance? Your body doesn't feel it. It's irrelevant. 10 megahertz is irrelevant. But what I'm suggesting is coming from the other way. Instead of a natural occurrence, if you can stimulate from outside the body, maybe you can do different things within the body. We're a long way from that. Admittedly, we're all very fundamental. We're just trying to understand. Okay, how we can do this is another story. Getting things in, we may have to implant our chips or things like that, right? But maybe as a cell engineering platform, that might also work. So it's not about doing things at the natural frequencies, about artificial stimulation.
Right.
Yeah.
Now, I think another usage of your technique, because you show this sound stimulation can increase uptake of siRNA.
Yeah.
Yeah. Because we found very difficult to deliver sometimes certain this siRNA.
Yeah.
And normally we use electroporation.
That's right.
And about 50% of cell will be killed.
Yes, that's right.
Yeah.
Yeah.
So you think that your approach, normally do not kill any cells, or they are
Well, you kill some, but it's
but
still
you-
90. At least we've always seen 90% viability.
that's very good.
Yeah. So as a cell engineering platform, maybe there's potential, right?
Yes.
Ex vivo kind of
Yeah.
re-engineering.
Yeah. I hope I can discuss with you
Yeah, yeah,
more.
for
The
sure.
afternoon we had a
Yes,
meeting. We are
yes.
going to have a meeting.
Oh, cool.
That'll be good. Yeah.
Yeah.
Thank you.
Hi, Professor Yeo. Thank you for your very fascinating talk. So my question is regarding whether your group, whether you guys have observed any changes to mitochondria and the energetic metabolism of the cell, given that mitochondria is, I'm kind of thinking it's a little bit like endoplasmic reticulum, and calcium uptake do help. So I'm just curious. Thank you.
The short answer is we suspect we are. We just haven't got there in looking at it yet. Yeah, like I don't know whether you're here. At the beginning of the talk, I said I had all this work is done by one person. So she's getting there. But yeah, we just don't have the bandwidth. But we're starting to look at mitochondria. We're starting to look at the nucleus and things like that. And we see changes. It's just that it takes a long time to try to figure out what's going on. But happy if that's interest. It's something we want to look at, because the energetics will naturally change as well.
Sorry, if you don't mind, second question, kind of a little bit unrelated, but sort of related in a sense. I'm actually working in the field of microbiology, so I was wondering if you think this sort of phenomena could also be applicable to microbes, and I think that's where also the mitochondria thing was like.
Yeah, I've thought about this. Again, it's something that we're interested in, but haven't had the chance to do. It's a bit different because bacteria have a different membrane structure, right? Yeah. So I don't think some of these mechanisms apply. But yeah, I'm sure something will happen. I just don't know what will happen. And it's like, it's just, if we had all the resources in the world, we'll be looking at it, but we haven't got there yet.
Thank you.
Yeah.
So, Percy, thanks for your sharing. So, I really like that you feel bored and make some very interesting discovery. So, I probably have 3 questions, maybe. Mm-hmm. So, I wonder for discovery, is the way of the stimulation very critical? For example, if you use just a ultrasound transducer and just shine on the cell, does it will it show similar phenomenon? And second question is, do you have any idea why like this magic number of like megahertz differentiate the behavior of the cells?
Yep.
And another question is, I think you emphasized that you use very brief stimulation. So so we do some neuromodulation using lower frequencies ultrasound. So we use brief stimulation as well. So do you think that really would show something similar or in that sense?
Yeah. Let me answer the first 2 questions together because they're related, right? So I'm beginning to think that frequency doesn't matter, and this review paper talks about some of this. In terms of what you how it doesn't matter, the mechanism and all. Where frequency matters is how, I guess, and this is why we only stimulate for a little while, or the outcome of why what happens when you stimulate for a little while. Because when you stress the cell too much, you push them down the apoptotic pathway, right? So you can only do so much. You can only get so much calcium in the cell, it dies, right? So because we use high frequency, we don't, and its short-term stimulation is very gentle. We don't kill the cells. We can modulate a lot more, right? So some effects that we see, you can't do because the cells already died with the low frequency. And because you have heating, you have cavitation, it's very, very stressful on the cell. So that's where it matters. So I hope it makes sense that generally it shouldn't matter, but the way you don't see some of these things because at lower frequencies the cells die before you can even do anything. Okay. Now maybe that also may answer the third question a bit, right? Now, with neuromodulation, I suppose I won't pretend to know. But I suppose it depends what you want to do. I guess if you're matching action potentials and things like that, there's a sweet spot in frequency. So I won't pretend that the megahertz, is the answer to everything. But I think we're starting to look at neuromodulation a bit. There's a whole field of sonogenetics, right? And this activation of I think some certain ion channels has related to neuromodulation and all the genetic changes that. So I think there is because if we can activate those piezo channels or something like that, or even maybe beyond piezo channels, the TREC or the TRIP channels, things like that, we can also drive things from the other side. So again, it's a different way of looking at things, maybe doing different things.
Thank you.
Thank you, Leslie, for the exciting talk. I have a quick question. So my experience with SAW was about 7 to 8 years ago.
Oh, wow.
So I don't have an updated understanding of how the technology is right now. So in the past, my friend David had to carry this really big machine to generate the SAW waves. Is it still like that now? Because you discussed like it is the potential to implant in vivo for clinical implications. Just wondering if we can actually miniaturize it enough right now for such integration. Thank you.
You're right smack bang on the buck. You want to translate some of these, you have to move away from your signal generators, your amplifier switch. Are not only big, but they cost a lot of money, right? So we're trying to build something small. It was for a different application where we're trying to, we have a nebulizer that can deliver biologics using the acoustic waves. So we're trying to build handheld portable nebulizers. So we're trying to shrink everything. So it's possible. And this is any electrical engineers here. I mean, this is where we need some help because this RF space is very unique. 10 megahertz. Okay, so I tell you it's a mishmash of all the problems, right? So at least for nebulization, not this application, but nebulization, we need a lot more power, right? So it's high power and high frequency, 10 megahertz, okay? So when people do electronics, you know, your computers or your mobile phones are up in gigahertz. It's very low power, okay? And then you've got people who do, you know, the conventional RF, which is all, you know, the conventional ultrasound, for example, is all down there, low frequencies, you know, low powers or high power. So we're in this space where very few people work with. So this is the challenge, you know, trying to design circuitry and, you know, components in this very unique RF space. So we're slowly getting there, but, you know, I'm not an electronics engineer, so if there's anyone here who knows how to do these things, you know, we appreciate a lot of help. But that's the goal. It's getting smaller and smaller, but, you know, trying to design circuits. I think for this application, you know, some of these things which have low power, it can be done. It's the high power ones, the nebulization, that we're struggling a bit with.
Thank you.